Part 2 of Chapter 12. Now, there's a segment on DNA profiling. This covers sections 11 through 16. DNA profiling is used in all kinds of things. It's used in disease diagnosis, it's used in forensics, it's used in paternity disputes, and it's even used in basic science in terms of comparing DNA from, say, Neanderthals to modern humans. Now, in section 11, it discusses PCR. PCR stands for polymerase chain reaction. Now, in many cases, a DNA sample may be really small, not enough to get a good analysis of it. So if one can make many copies of a DNA sample, many identical copies, then there can be enough quantity to sample it properly. So polymerase chain reaction uses DNA polymerase to make multiple copies of a set of DNA so that you can amplify your sample. Polymerase chain reaction, again using DNA polymerase, it's the same enzyme that is used in our own cells when we replicate our DNA. Now, the way to analyze the DNA is by gel electrophoresis. Now, with gel electrophoresis, you have the ability to load DNA samples in a gel. Now, this gel is, a little bit bigger than a stack of cards, just a few millimeters thick. And it's made of a gel-like substance, and it's laid down flat in a special electrolyte solution. And the DNA samples are loaded in these wells. This thing lays flat and is subject to an electric current. Gel electrophoresis separates molecules based on their size and their charge. DNA, generally being negatively charged, is going to be pulled toward the positive end. So they all start up here. You turn on the gel, and the segments migrate down the gel over time. Now, of course, the smaller the segments, the faster they move. So when the gel is done and stained, you can see these bands and you can compare samples. Now, each band represents a segment of DNA of a certain size. They can all be compared to the known sizes of a standard, and you can also compare samples to each other to try to match them up. Now, STR stands for short tandem repeats. These are segments of our DNA that are highly variable between one person and another. They're almost like molecular fingerprints. So if you're going to compare DNA in terms of forensic testing to try to identify a person, you don't want to use the DNA that's the same in everybody. You want to use the DNA that's highly variable, very different from one person to the next, like a fingerprint. Now, RFLP analysis, RFLP stands for restriction fragment length polymorphisms. There's that word again, restriction enzyme. So what you do is you take your DNA samples, you cut them up with the same restriction enzymes, which means that you're going to have all these pieces of DNA cut up, but they're all mixed together. So you take your cut up DNA and you run an electrophoretic gel, and these would represent restriction fragments, pieces of DNA after they were cut. Now, if you're using highly variable regions of DNA, then you'll look at matching patterns. Now, you'll notice that the only two samples that match are samples B and D, A and C being different. So not only can you match samples from the same person from different locations, but you can also look at the actual sizes, because when you run the standard, these are fragments of known size, so that this data can be compared to other electrophoretic runs in other, say, forensic cases. So that's a little bit on DNA profiling, forensics, etc. Now, the last sections, sections 17 and 18, discuss genomics. Genomics is the study of all of the DNA in an organism. Now, of course, we talk mostly about the human genome. The Human Genome Project's been going around for a number of years, but we've learned quite a bit in recent years. For example, how big is our DNA in our cells? Well, your DNA in each of your cell nuclei consists of 3 billion base pairs. Now, that's per haploid set. So when you're talking about diploid, you double that, okay? Of that, about 21,000 gene loci. Yeah, just 21,000, okay? 21,000 pairs of alleles to work with in a given organism. But that's not all of your DNA. There's a pie chart in section 18. Only 1.5% of our DNA is actually transcribable. In other words, 1.5% represents these gene loci that are actually transcribed into RNA and then traits. But what about all the rest? Well, some of it is simply non-coding sequences. Some of them are repeated sequences, and some of them are regulatory sequences, such as promoter regions and termination sites and so on and so forth. So be sure to study your notes concerning these, and this concludes the review.